Hi everyone, and welcome to the first chapter in this Implementing AlphaFold 3 from Scratch series. Today I'll show you how you can implement the full feature extraction pipeline from AlphaFold 3 yourself. So in the video, you'll kind of see the theory on how you do it, and then afterwards you can go to the link in the description to the GitHub repository and do the full implementation yourself. Feature extraction is the process of turning the domain-specific data of a field into the common format for machine learning, tensors. This step can be kind of trivial in other domains, for example in image recognition, where images basically are already tensors. In biology, however, that is not the case. Biology has a lot of quite heterogeneous data, and the first step in any BioML project is to think about how you make this data accessible. In AlphaFold 3, we use structural protein data from the PDB and genetic data from a genetic database in the form of a multiple sequence alignment. Directly accessing biological databases like the PDB can be quite hard, and that's why I'm particularly happy that the lab of David Baker, whom you might know from AF diffusion or protein MPNN, released the AtomWorks framework in the last year. AtomWorks tries to be the bridge between you and the databases, so that working with biological data becomes simple. It is kind of like infrastructure for BioML, and I think this could be one of the steps that really brings up the pace with which computational bio evolves. In fact, we could have basically skipped this chapter and started with the next one, because AtomWorks already has a fully fledged AlphaFold 3 pipeline. Unfortunately for you, we aren't gonna make use of that, mostly because the whole thing about this series is to explore how everything works, but also because I want the final model to work with the AlphaFold 3 weights, and for that, our feature extraction process needs to be 100% equivalent to the original AlphaFold 3 implementation. AlphaFold 3 has some quirks there that AtomWorks doesn't fully cover. However, we'll make use of individual transforms of AtomWorks and its capacity to interface with the PDB, especially for the fifth part of this series, where we'll be trying to train AlphaFold a bit. Now, let's get into the details. AlphaFold 3 has three main groups of features: token features, reference features, and MSA features. Aside of them, there are the template features, which we won't implement, we'll do template-free prediction, and the bond features, which are pretty simple and which we'll talk about in the end. Generally, the token features will have shape number of tokens C, the reference features have shape number of atoms C, and the MSA features have shape number of MSA sequences and tokens and C, where C are the channel dimensions. Here, a token is a group of atoms, for example all the atoms belonging to one residue. We'll make this more precise in a moment. Throughout AlphaFold, the token features will be turned into relative position encoding, similar to how we would encode the position of a word within text for a large language model. The reference features are processed by the atom attention encoder, which wrangles the atom dimension into a token dimension, and the MSA features are entering the model through the MSA module. These steps will be exactly what we'll do in the next video. Now for the token features. As said, a token corresponds to a group of atoms. Concretely, for standard proteins or nucleic acids, one token corresponds to one residue, that is, one amino acid or one nucleotide. Ligands or modified residues are atomized, meaning they are encoded using one token per atom. For the example of phosphoserine here, we'd use 11 tokens, since we are ignoring hydrogens in the model. These tokens are the fundamental unit of the machine learning model, meaning that all core tensors within the model have this as their dimension, and operations like attention are carried out along this token dimension. The reason that we are not just using one token for one atom everywhere is to reduce the computational complexity of the model. This here is the data type for the token features of AlphaFold 3. We have a number of indices and IDs, an encoding of the residue types, and flags for the type of token, meaning whether it is part of RNA, DNA, a protein, or a ligand. We'll go through the features one by one. First up, we have the residue index and token index. Token index is basically just a range cutting up from 1 to the number of tokens. Residue index counts up starting at 1 within the residues of each chain. The NDG shown here is the CCD code of the ligand N-acetylglucosamine. Within ligands, the residue index is just 1. The names DA and DG in the example are abbreviations for deoxyadenosine and deoxyguanosine, two of the DNA bases. SMID, Entity ID, and SIMID are IDs on a chain level, which means that within a chain they are constant. SMID gives different IDs to each chain. Entity ID is shared between identical chains, so it would be the same for two subunits in a homo-oligomer, but different for two chains without identical sequence. SymID gives a distinction for chains within an entity ID. Within each group of chains with the same entity ID, it counts up from 1 to the number of chains in that group. We can see that here for the chain glycine-glycine-serine. They are identical, so they have the same entity ID. Within this group, they are enumerated by SymID as having SymID1 and SymID2. The mask of the token features is just identical 1 for each token in the input. It only gets entries that are not 1 when the input is padded, for example to stack multiple inputs in a batch that have varying length. The res type feature encodes the residue types of the tokens, with unique integers for the different amino acids and nucleotides. Concretely, we do an encoding with 32 classes. The 20 standard amino acids, a class for the unknown amino acid, a class for the gap symbol in MSAs, and for both RNA and DNA we have 4 classes for the standard nucleotides and 1 for an unknown nucleotide. Just to warn you, this is not exactly the order that AlphaFold 3 uses. In AlphaFold 3 they have different encoding orders and different number of classes for encoding of the res type and encoding of the MSA, since you don't need the gap for the res type here. In our implementation I unified that so that it is a bit more consistent. That works because I can just switch the columns of the weights when loading the original AlphaFold parameters into our model. The last 4 token features are simply flags that state the type of the tokens, that is, whether they belong to DNA, RNA, protein, or a ligand. These are only created for convenience during feature extraction and don't actually make it into the features that are used in the model. AtomWorks is quite convenient for computing these features. After loading a PDB entry or a sequence with AtomWorks, it is represented as a so-called atom array. An atom array is basically a set of named NumPy arrays of shape number of atoms. It contains annotations such as atomarray.resID, atomarray.resName, or atomarray.atomName. These are just NumPy arrays, so we can index them with all the standard techniques, slicing, Boolean indexing, or integer indexing. AtomWorks also provides some convenience methods, such as getTokenStarts. This computes the indices where the different tokens start in the atom array, based on the rules we discussed. 1 token for a residue, a nucleotide, and 1 for each atom in an atomized structure, meaning a ligand or a modified residue. The first glycine has 4 heavy atoms, so the second token starts at index 4 in the atom array. NDG is a ligand, thus atomized, so the token starts only increase by 1 for these tokens. Using this, we can compute the token feature residue index from the atom array by computing the token starts and then indexing into the atomarray.resID annotation. We can also index into the atom array directly, which gives an atom array with this index applied to all its annotations. As another example, let's go through how we could compute SMID. In the atom array, this corresponds to the annotation PN unit IID. PN stands for polymer non-polymer unit, and AtomWorks uses this to be more specific about how it handles SMIDs for non-polymer structures, such as ligands. If we retrieve PN unit IID from our token array, it might look like shown here, with different strings for the different chains. Now, to encode this with numerical IDs, we can use NumPy's unique function. It calculates the set of unique IDs, and also which indices in the array have which of these IDs if we set return inverse equal to true. These indices have exactly the desired property of SMID, that they identify the different chains, meaning chains with different PN unit IID. For our last example, we'll look at SymID, which identifies the different chains within a group of identical chains. To compute that, we can iterate over the different entity IDs, which mark the different groups of identical chains. Then we can create a mask that selects only the tokens with this entity ID. Within this group, we can use np.unique on the PN unit IID again, as we did for the SMID, to get different IDs on different chains. Then, finally, we can use the mask on our output feature SymID and set these SymIDs to the positions belonging to that entity group. I know indexing can be a bit tricky to wrap your head around, so feel free to stop the video for a moment to check that this makes sense for you. As a last technical note, AlphaFold 3 rounds the number of tokens up to some fixed values, like 256, 512, or 768, and so on, and pads the features accordingly. We'll call this padded size the token count, or explicitly the padded token count, and the original one the unpadded token count. That's it for token features, now to reference features. These are per atom features, and most of them pretty much directly correspond to properties in the atom array. This here is the data type we'll be implementing for the reference features. Charge and element can be obtained from the atom array without any changes. The atom names are padded to length 4 with spaces, encoded using the ASCII numbers, and then reduced by 32. Because a space has the ASCII encoding 32, this way spaces are mapped to zeros. Note that we are ignoring hydrogens. AlphaFold doesn't predict them, and they are not considered in the features either. Further, the reference features include the atom's token index and ref space UID, meaning the index of the token it belongs to and the index of the residue it belongs to. We compute these by first getting the indices of the token and residue starts, respectively, using the corresponding atomworks functions, and then just checking to which of these each atom position rounds down to. For the token indices, it would look like this. Here, the atom indices are just arranged from 0 to number of atoms minus 1. The token starts array lists the indices of where each token starts. In your code, you'll implement a round down to function that computes, given target values such as token starts here, and source values such as the atom indices, to which index each source value rounds down to. The last reference feature are the reference positions. These are the proposed three-dimensional structures of the individual residues and ligands, not in the global protein structure, but just for each residue on its own. We call these proposed 3D structures of the molecules a conformer, and we can use the computational chemistry library RDKit to compute them. We will use RDKit through AtomWorks, which means that this computation of the positions is basically just 3 lines. For a known CCD code, we first query AtomWorks for a molecule object of that CCD code, which is the internal representation RDKit uses for molecules. Next, we generate conformers for that molecule, and in the last step, we convert them from the RDKit type molecule to an atom array, which is what we use in AtomWorks. For molecules that are not in the CCD, we can basically do the same thing, but we don't construct the molecule object from the CCD code, instead from the data in the atom array, which contains all necessary information on bonds, elements, and so on. Note that we are not taking the 3D positions from the CCD, not even if the CCD would contain some. We just construct the molecule object based on the CCD, which only uses bond and element information. The conformer in each case is generated by RDKit through a computation. After that, we just plug these positions into our feature. Note that we are reusing computed positions of the same residue within a chain. So if a chain has 2 serine residues, we'd only do one call to the conformer generation, store the coordinates, and use them for both. If there's another serine in a different chain, however, we'd recompute them. Just as with token features, AF3 pads the atom features to a shape of number of tokens times 24, using a maximum of 24 different atoms per token. Again, we'll call this the atom count and refer to the real number of atoms as the unpadded atom count. These are all the reference features, but there's one more thing to talk about regarding them: the conversion between the atom layout and the token layout. In the atom layout, the features are simply in one long tensor. In the token layout, however, they are split into different rows, corresponding to the tokens they belong to. Features in atom layout have shape number of tokens times 24, and features in token layout have shape number of tokens 24. However, the conversion between them isn't a simple reshape, and that is because the atom layout is dense, while the token layout has gaps, because not every token is using all 24 atoms. Still, conversion between these layouts is pretty easy. We can just create a mask for the token layout, which atoms are present, and the same mask for the atom layout. For the token layout, this mask can be constructed through a broadcasted conversion of a range from 0 to 23 and the number of atoms each token has. It's good torch practice for you to think through why that works. Then the conversion of a feature can be done with Boolean indexing, as out of token layout mask is equal to feature of atom layout mask, or vice versa. One thing that I think is kind of funny is that DeepMind actually does this conversion here wrong at one point, where they accidentally do a simple reshape, which kind of messes up their input embedding. But we'll see that in the next chapter. The last group of features are the MSA features. As you might recall, an MSA is an alignment of many sequences that are similar to the target sequence. I've only shown one sequence here for simplicity. The alignment process can introduce gaps in both the target and the align sequence. Gaps in the align sequence are marked by a gap token, which I represented by a dash here, and gaps in the target sequence are stored through a deletion count array. This array for each residue in the target sequence states how many gaps there are in front of it. In this example here, for the alignment of the target and align sequence, we get a 2 in front of the K. This number is called the deletion count in AlphaFold, or the insertion count in AtomWorks. You could say that from an evolutionary point of view, these are residues that were deleted when mutating from the aligned to the target sequence, or inserted when going from the target sequence to the aligned sequence. So it is a matter of perspective whether you call it a deletion or insertion. Notice that in the original alignment, the length of the aligned target sequence varies depending on how we aligned it, because it might contain additional gaps. Through the transformation that we performed, the length of the aligned target sequence is exactly the length of the original target, because we stored the deletion count in a separate array. This same size property is very convenient for us, because now each MSA entry has the same number of columns, although we lose the information on which additional amino acids were present in the aligned sequence. AtomWorks provides us with these deletion counts and an integer encoding of the MSA residue types, including a special token for gaps. Because of that, unlike in the AlphaFold 2 series, we won't need to compute them ourselves. To derive the necessary features from them, we have to go through 3 steps. In the first step, we encode each residue in the MSA with an integer, using the same encoding we used for the rest type feature. In the second step, we concatenate the MSA encodings of all of the individual chains into one large tensor. And then, third, we do some calculations on that tensor to build the final MSA features. So, first step, MSA encoding. AtomWorks actually already provides the MSA tensor as an integer encoding, but it uses the wrong integers. To fix that, we just need to apply a map to the AtomWorks integers. If AtomWorks encodes glycine as a 3 and we want to encode glycine as a 7, we need to map all 3s to 7s. We can compute that efficiently using a lookup table. Basically, we can just create a lookup table such that lookup at AtomWorks ID is equal to AlphaFold ID for corresponding encodings, and then use integer indexing to transfer the encoding as lookup at AtomWorks encoding. With this indexing, AtomWorks encoding can have any shape we want, and it works because of the rules of integer indexing. The second step is concatenating the MSAs of the different chains. This is done as follows. First, we create a fixed size all zeros tensor of shape max MSA sequences and tokens. Max MSA sequences is a constant and independent of the input. In the top row, I indicated which chains the different sections of the tokens belong to. We set the block max MSA size and token to the encoding of the gap token. Here, max MSA size is the number of rows in the largest MSA that is in the input, which would be the one of C1 in this case. The first line of the feature is set to the token feature arrest type. Then we iterate over the chain IDs that have an MSA available, and copy that into the corresponding region in our big MSA feature. Note that it doesn't matter if we overwrite the first row or not, since the first row in an MSA is always the target sequence, so the segments in the MSA encoding are identical to those in the rest type feature. One thing to watch out for is that the MSA has as many columns as that chain has residues, while the number of tokens for that chain can be bigger in case a residue was atomized because it was modified. In the example here, phosphoserine is a modified residue and thus atomized, which means it is encoded by 11 tokens corresponding to its 11 heavy atoms. In the graphics, I'm only showing 3 tokens for it. The MSA for this chain, however, only has 4 columns, so we have a shape mismatch. In that case, we put the corresponding column from the MSA only to the first token belonging to the residue. The full MSA concatenation process, just as we did it for the MSA encoding, is repeated for the deletion count. The last step performs some calculations on the deletion count and MSA and assembles the final features. Concretely, we calculate the following values. First, the profile. For each token, the profile is the distribution over how often each residue type occurred at that position. So it's of shape number of tokens 32 for the 32 different classes we used during encoding. A straightforward, although not super efficient way to calculate this distribution is by doing a one-hot encoding of the MSA and then averaging over the number of sequences dimension. One thing to keep in mind here is that for the mean calculation, we need to divide not by the feature shape in that dimension, but by the number of rows actually present for this residue position. So we'll do that with a masked mean operation. Second, the deletion count is transformed into 3 values. Has deletion, which is 0 or 1, to indicate whether deletion count is bigger than 0 at that location. deletion value, which is computed as 2 over pi times arctan of deletion count over 3, and the deletion mean at each location. Now we assemble the features. The MSA features, constructed as the concatenation of the MSA1 hot encoding, has deletion and deletion value. The second MSA-based feature, the so-called target feature, actually doesn't have an MSA dimension because it contains only row-wise average MSA features. It is created by concatenating the one hot encoded rest type token feature, the MSA profile, and the deletion mean. The MSA feature, the way we reconstructed it, is a gigantic array. Its leading dimension is the maximum allowed number of MSA sequences in AlphaFold 3, which is about 16,000. Because of that, AlphaFold doesn't use the full feature, but samples uniform random subsets of the rows, consisting of only 1024 rows. If you did the AlphaFold 2 series before, the sampling here is quite a bit easier than what was done in AlphaFold 2 to reduce the MSA feature size, where we selected special sequences as cluster centers and assigned non-cluster sequences to centers for aggregation. As we'll see in the next video, the AlphaFold 3 model runs over multiple recycling iterations, and for each we sample such a subset independently. For an iteration over 11 cycles, for example, we'd construct 11 MSA feeds of shape 1024 token count by sampling rows from the full MSA feature. We are almost done. As the very last feature, we have the bond matrix. It is basically a Boolean matrix encoding which tokens have a covalent bond between them. Note that it doesn't indicate bonds within a residue or between standard residues. A bond between tokens I and j is only marked if at least one of them is atomized. Also, only the upper triangle is filled out, so only bonds between tokens with indices I less than j. Through these rules, the bond matrix only shows bonds within ligands or modified residues and interchain bonds. AtomWorks already provides the covalent bonds through atomarray.bonds. The only thing we need to do is to go from the atom indices in the AtomWorks array to token indices, which we can do with our by now well-practiced trick of computing the token stats within the atom array, then rounding the atom indices down to them and getting the index of the token they were rounded down to. With that, you know everything you need to implement the feature extraction pipeline yourself. The documentation within the tutorial files is a bit more explicit on some operations and gives you hints on how to actually implement it. One thing I want to make you aware of is that there are some operations in the code that I implemented for you already, which are prefixed with hotfix. These take care of some quirks in the AlphaFold 3 implementation for which I don't see any real reason. These are not big things. Some examples would be that AlphaFold 3, due to the way it handles MSA pairing, duplicates the first row in the MSA in case there is only one chain that is provided in alignment. Or that AlphaFold switches the token used for ligands in the rest type feature, the unknown amino acid token X, to the gap token in the MSA feature. So, when you see a transform named hotfix something, you can read through the code if you feel like it, and apply it at the point the documentation tells you to. So, that's everything from me for today. In the link in the description, you can get to the GitHub repository, where you'll find the tutorial code and instructions on how to do the setup. I hope you have at least a bit of fun implementing the feature extraction. I think it can be a bit of a pain to do that, but I think it's still quite important to also do the feature extraction step, because it's how you access a new domain and make it available for machine learning. Still, if you get annoyed and want to stop doing it, for this video you've got my permission to just go over to the solutions folder and copy the code from me over to your tutorials folder, if that's what's holding you up from going on with the series. So with that out of the way, I'd love to hear some feedback from you, how you liked the tutorials, if they were too difficult or if they were too easy and I babysitted too much. And in general, I'd just like to know if some of you completed them. So please write me in the comments, or if you see any issues with the code, you can also create an issue on GitHub. And with that, I'll say goodbye for today and see you in the next video.